Thanks for the kindly introduction. Good morning, good afternoon, and good evening. Welcome to the MAQC Society webinar series. Today, I'm going to present a piece of work done by the SEQC2 OncoPanel sequencing working group. In this study, we assessed the performance of the variant calling with targeted deep sequencing through a multi-lab cross-oncopanel study. We found that the high sensitivity and reproducibility can be reached if the evaluation is done within targeted regions and within certain allele frequency ranges. Please allow me to read the disclaimers. This research includes contributions from and was reviewed by the FDA and the NIH. This work has been approved for publication by these agencies, but it does not necessarily reflect official agency policy. Certain commercial materials and equipment are identified in order to adequately specify experimental procedures. In no case does such identification imply recommendations or endorsement by the FDA or the NIH, nor does it imply that the item identified are necessarily the best available for purposes. First, I'm going to briefly talk about our motivation. The precision therapy and the prognostic prediction for cancer patients are big components in the precision medicine era. The advantage of technologies, including the next generation sequencing, the OncoPanel testing, as well as the bioinformatics tools, bring hope to the patient that they may receive targeted therapy rather than general treatment when genomic indicators can be found with these technologies. Just like many regular medical examinations you may have when you visit a doctor, for example, the body weight and the blood pressure, the molecular testing requires accurate and reproducible results. I still remember 20 years ago when I was doing my internship in a hospital that every morning I measure the blood pressure for my patients. I need to measure 3 times and put down the average values on their medical records. The American Heart Association recommends at least 3 resting measurements on at least 2 separate healthcare visits to diagnose hypertension. Multiple testing and having the consistent results are the keys for accurate diagnosis. However, in many cases, the patients may only have a single test. For some instances, it is totally fine to test just once because the measurements are reproducible and the variation is small, like my nurse only scale my body weight 1 time for each of my doctor visits. If a test can be done only once, the test needs to be highly reproducible, no matter it is reproducibly right or wrong. Reproducibly wrong is not always a bad thing, especially when the errors can be fully aware of and eliminated. Reproducibility can be further explained by sensitivity and the false positive rate. Sensitivity, also called the true positive rate, measures the proportion of positives that are correctly identified. Of course, high sensitivity leads to high reproducibility, while randomly occurred false positive will lower the reproducibility. In the clinical practices, people are more focused on high sensitivity because once specific biomarkers can be identified, the patient may be able to receive focused therapy, which are believed to be better choices to save lives. However, as a trade-off, high sensitivity may also lead to high false positive rate, which is somehow overlooked. False positive detections will also increase worries or anxious. In a letter to healthcare providers last year, the FDA was alerting clinical laboratory staff and healthcare providers that false positive results can occur with COVID-19 antigen tests. Another study found that the patients who receive a false positive mammogram result are also prescribed anxiety or depression medication at a rate 10 to 20% higher than patients who receive an immediate negative result. With this said, how to get reproducible and sensitive results with the false positive rate to be controlled to a low level is a very important question to be answered. In recent years, FDA approved or cleared several cancer panels which are designed to provide physicians with clinical actionable information. This is a great movement in the precision medicine era that a minority of cancer patients start to benefit from the molecular tests and receive targeted therapies. However, these cancer panels have some limitation. Generally, only a small number of biomarkers are covered by these panels, and the minimum variant allele frequency that the panel testing can provide is limited. Some panels also claim that they could be used for tumor mutational burden estimation, but the estimation is usually based on a small number of genes. Pan-cancer panels, on the other hand, cover a large number of genes and can detect mutations with a variant allele frequency as low as 1% if given sufficient sequencing depth. The pan-cancer panels are currently still for research purposes. Before they can be adopted in the clinical tests, a thorough understanding of their reproducibility, sensitivity, and the false positive rate is greatly needed. So with all these things in our mind, a large number of members from the MAQC society, the government agencies, the industry, and the academy came together and formed the SEQC2 OncoPanel sequencing working group. We made this study design try to address the questions we just asked. If you attended the webinar last week, you may remember that we developed reference sample A and the sample B. The sample A is a one-to-one mixture of 10 cancer cell lines from the adjuvant UHRR material. Sample B is a cell line derived from a normal male individual available at the adjuvant. So we also developed the sample C and the sample spike-in. The sample C is actually the one-to-one dilution of sample A and B. And the sample spike-in is a mixture of sample B with 5% acromatrix hotspot synthetic controls. We distributed the 4 samples to 8 participating panels, including Agilent, Burning Rock, IDT, iGeneTech, Illumina, QIAGEN, Roche, and Thermo Fisher. We asked each panel providers to recruit at least 3 test labs which are well established for their panels. Each lab used the panel to make 4 library replicates for each sample following the harmonized experimental protocol by the panel provider accordingly. In total, we have 28 test labs and 430 libraries. The libraries from the same panel were sequenced on the same sequencing platform, including Illumina HiSeq 2500, NovaSeq, NextSeq, and the Thermo Fisher Ion Torrent S5. So each provider performed the downstream analysis using their recommended or in-house bioinformatics pipelines for variant calling. Variant calling results were later submitted in a VCF format, along with their reporting regions and the whitelist, blacklist to an independent group led by Dr. Joshua Xu for the performance evaluation. For sensitivity, we rely on the known positive set from the verified genomic reference sample presented last week, webinar by Dr. Wendell Jones. For the false positive rate, we developed 3 methods. We'll talk about the 3 methods later. And also, we evaluated the reproducibility, the intra-lab and the cross-lab reproducibility. So here's some basic information about the 8 participating panels. All the panels are currently for the research purpose only and are commercially products available on the market, except the Roche panel is a custom designed for this study. Each panel have different panel size. From the biggest one is 7,600 kilobases, and the smallest one is about 150 kilobases. And all the panels have a different input amount requirement, and the samples were sent to different sequencing platforms. And also the panel provider run their pipelines and with certain VF cutoff. So more detail, you can find more detail of the panels and the whole study and the results in our to be published manuscript in the genome biology. So to achieve our objectives, we evaluate the panel performance by estimating sensitivity, false positive rate, and the reproducibility. We evaluated the performance in different VF ranges. We mainly focus on the SNV, but we also take a look at the MNVs and small indels. We also try to understand the impact of variant allele frequency cutoff and the different genomic regions. We also discussed the applicability of using the OncoPanel to estimate the tumor mutational burden through a simulation approach. Of course, there are some other factors which could impact the OncoPanel performance, such as the DNA input amount, sequencing depth, and other wet lab procedures. Considering these settings should be packed as part of the OncoPanel, we let the panel provider to harmonize the experimental protocol for their panels. We also let the panel provider to run their recommended or in-house pipelines as the ready-to-use bioinformatics pipelines. These pipelines were developed by the panel providers, some of which are also provided along with their OncoPanel products and should perform well for their panels. There may be better choices of pipelines or tools or parameters, but we decided to save ourselves from making the study too complicated and set this challenge aside for later. If you are interested in this topic, you probably can get some of the ideas from the webinar on February 23rd this year, presented by Dr. Wenming Xiao, who led the SEQC somatic mutation working group. Their study is focused on the whole genome sequencing. But you still get some ideas. We also try to save ourselves again by staying away from the complex variants such as long indels and the copy number variations and other structural variations. Now here are the results. First, we evaluated the sensitivity with the non-positive set. Since the non-positive set was reported within the consensus target region, especially for the class 1 non-positives. We had to limit the evaluation within the CTR region and within the panel reporting region, of course. I leveraged this figure from Dr. Windows Jones' presentation last week to showcase, and here is the consensus target region, shortly CTR regions here. It's the overlap of the 4 exon panels and also overlapping with the high confidence regions and remove the low complexity region. So we have the non-positive set for the CTR, and we can use them to see how many of them can be identified by each panel. Overall sensitivity across all the VF ranges in our panels was high. You see most of them are green. if I remember correctly, all panels claim that they are good for VF above 5%. So if you look at the 5%, they probably all green is good sensitivity. We also looked a step lower and find the sensitivity is still good for the lower range from 2.5 to 5%. The sensitivity dropped significantly in the 1 to 2.5%, which may be due to the artificial VF cutoff. Although the sensitivity looks pretty above 2.5%, but it's not perfect. We also took a close look at those few false negatives and found that most of them can be explained by a combination of low VF cutoff and the except for the Illumina in sample C for this region, there's one single false negative in all library replicates. We reported that. We were expecting perfect or almost perfect sensitivity from the sample spike-in because the spike-in variants are synthetic controls. But in some of the panels, the sensitivity is not as high as we expected. So we dived into this issue and figured out that the low sensitivity in the sample spike-in some of the panels is due to the interference of the nearby spike-in variants with the amplicon primers or the DNA capture primers. So all due to the high dense the variants that make a cluster. So both the spike-in variants and the variants from sample A or sample C are so close and therefore it's too difficult to correctly identify. So my personal comment here is that if you're going to using a spike-in, so and then you need to make the right use of it to be aware of some of the close to each other variations. We then estimated the false positive rate for sample A and the sample C. We have the known negative set for the CTR, and well, the known negative set is also not a complete set. We simply estimated our known negative set is about 50% of the total negative positions. So that is why we also developed the other two methods, try to estimate the false positive in different perspectives. So the method A is using the known negatives. Method B is we take the sample B, and because the sample B is a normal sample, so we consider all the variants below 10%, from a 1% to 10%, they are considered as false positive. And also, the other method is because sample C is a mixture of sample A and B, so every the variants identified in sample C, but not in A or B, were considered as false positive. So we just take the simple average of these three methods and draw this plot. We found that the highest false positive rate is about 10%. All other for all the panels, and all other panels are below the 10, not the 10%. Sorry, it's the 10 per million base, 10 false positive per million base. But when you lift the VF cutoff and to like 5%, so all panels have a similar false positive rate of about 1 per million or even less. All panels report no false positive cause for the VF above 10%. So sensitivity and the false positive rate requires the true set of positives and the negatives. Well, if the true set is not available, which may be the reality in many cases, we still can do the reproducibility estimation. As I have discussed earlier, both the sensitivity and the false positive will contribute to the reproducibility. We calculate the intra-lab and the cross-lab reproducibility for variant cause in the CTR regions with VF between 2.5% and 20%. And we found that the intra-lab and the cross-lab reproducibility were quite similar for each panel. Besides the cross-lab reproducibility being slightly worse than the intra-lab reproducibility in a few instances like these. We also observed the noticeable lower reproducibility in the VF range of 2.5 to 5%, which may be due to the VF cutoff by the panels. Remember that the independent labs were recruited by the panel provider, who made sure that they were well established for their panels and followed the harmonized experimental protocols by the panel provider. So the good intra-lab and cross-lab reproducibility is expected. So we believe that some sort of the certification and the regular internal quality check is very necessary for generating reproducible result, and so for the clinical testing. Although we mainly focused on the performance assessment on the SNVs, but other types of variants may also be interested. The total number of small indels and MNVs for all panels is too small for regular statistical analysis, but still worthwhile for a glance. For some panels, the small indels and the MNVs were identified in some labs and replicates, but not in others, which led to a lower sensitivity for the small indels and MNVs than the SNVs. Higher sensitivity was generally observed in higher VF ranges for both SNVs and the non-SNV variants. The number of small indels and the MNVs are relatively small for each VF ranges, but once there's a larger number of them for the evaluation, the sensitivity of the small indels and the MNVs are close to or slightly lower than the SNVs in the same VF ranges. There's only a handful of small indels and MNVs tested in the Thermo Fisher panel, where the sensitivity is lower than the SNVs. The samples for this panel were sequenced with the ion torrent platform, which is known to have the drawback on the indel detections if there's no optimization of the indel detection applied. Almost all the panels, all the variants calling pipelines need to set a VAF cutoff for reporting, and such an arbitrary cutoff will have some impact on the sensitivity. With a certain cutoff, the VF of a variant may be slightly lower than the cutoff and was not reported, so the sensitivity will drop a little bit. We call this situation as boundary effect. We did the full cutoff to see the impact and found that the sensitivity rose expectedly across most panels in all reference samples if the cutoff was more relaxed, where the boundary effect will be also decreased. No matter what cutoff we take in this analysis, if only taking the known positive set with the VF above 5%, the sensitivity is all great for all panels and for both sample A and the sample C. The boundary effect is greater in this 2.5 to 5% ranges and cause the noticeable drop of sensitivity. Our previous evaluation was done within the CTR. You may be interested in how the performance looks like if moving outside of the CTR. The class 2 known positive set allows us to take a quick look, but maybe not statistically sound due to the small number of variants outside of the CTR. If you want to know more about the class 2, please refer to last week's webinar. Dr. Wendell Jones talked about the class 1 and the class 2 variants in our reference sample. According to our observation, most panels performed consistently within and without the CTR, except these few cases, sample C from Illumina panel, Roche, and also sample A from Thermo Fisher, which show a slightly higher observed sensitivity outside the CTR, which is because the number for these cases, the number of known positive for these cases are extremely small, just a few of them. So we calculated the confidence interval for sensitivity using a permutation approach and find that the confidence intervals were narrower within the CTR, wider outside the CTR. At the beginning of my talk, I have mentioned that the high false positive rate, the false positive rate is somehow overlooked and may result in some bad things. For variant calling tests, the false positive call will lead to unnecessary or even wrong treatment and prognostic predictions. So in this study, we proposed the 3 methods for the false positive rate estimation. The method via KN depends on the known negative set and maybe underestimated the overall false positive rate because this is not a complete set. Method B, B low is considering any call in the normal sample B with the variant between 1% to 10% as false positive, but maybe overestimated the false positive rate by failing to exclude the rare mosaic variant caused by the cell line drift. Method C only count any variants called in the sample C as a false positive call if it was not called in any of the 4 sample B libraries with the VF above 10% or any of the calls from sample A. But this method also may underestimate the false positive rate by failing to recognize those false positive calls due to the systematic errors, as they would be caught in all 3 samples. So all the 3 proposed methods try to estimate the false positive rate from different aspects and have their own limitations. We compared the 3 methods and found that we are lucky they produce largely similar estimates within the CTR, except for the method below, in the certain VF ranges of IGeneTech and Illumina, the below method get a higher estimation than the other two. We checked on the false positive calls and found that almost all false positive calls within the CTR were irreproducible across library replicates for any panel sample and the false positive rate. So most of the false positive calls just happen once. They are not producible. They are just random false positives. So you may remember from our previous observations, most panels performed consistently well within and outside the CTR in terms of sensitivity. But for the false positive, we observed that the false positive rate is lower within the CTR than the outside the CTR across all the VF ranges and all the panels. We further partitioned the outside of the CTR regions into two parts. Outside the CTR, but within a high confidence coding and the rest. We observed that the false positive rate is the lowest in the CTR and the highest outside the CTR and outside the high confidence coding region, which is the rest. So this is a very interesting message. Maybe the sensitivity could be equally well within and outside the CTR. However, the false positive rate is different. And any application which requires the false positive estimation may need to take the CTR region to be considered. We also applied the different VF cutoff to see the impact on the false positive rate estimation. As expected, the higher the VF cutoff, the lower the false positive rate were observed in all panels. Remember, the sensitivity could drop with more stringent cutoff applied. And now we observed, and the stringent VF cutoff may reduce the false positive rate. So taking both results together, we consider a false positive, a VF cutoff of 2.5% may be a good choice for a trade-off between the sensitivity and the false positive rate. As we observed in that the false positive rate is higher outside of the CTR and the sensitivity didn't drop. There's no surprise that across most of the samples in both sample A and most panels in both sample A and C, the cross-lab reproducibility is marginally lower outside of the CTR, where the observed drop in the reproducibility may be driven by the more false positive calls outside of the CTR. As you may know, tumor mutational burden has shown potential as a predictive biomarker with several applications, including associations reported between different TMB levels and the patient's response to immune checkpoint inhibitor therapy in a variety of cancers. So if there's more mutations, and will result more the T cells recognize such cells, such many mutations. So more mutation identified, then there's it's kind of related to the the immune checkpoint inhibitor therapy. So this is very interesting. And on the June 16th last year, the FDA expanded the approval of immunotherapy drug. The drug name is difficult to pronounce, but there's a drug approved to treat any advanced solid tumor cancer with a TMB greater than 10 mutations per million base. So this marked the first time that the FDA has approved the drug with its use based on the TMB measurement. TMB is defined as the number of non-inherited mutations per million base of investigated genomic sequence. Broadly, whole genome sequencing, whole exon sequencing, and panel sequencing can all be used for the TMB estimation. Studies of the TMB from research perspectives typically using the WGS and the WES for the TMB estimation. However, for the clinical applications and the targeted sequencing, the panel sequencing is usually preferred because it's quicker and lower cost. As we just discussed, and if you remember, if you consider TMB, if the TMB is above 10 mutations per million base, however, if the false positive rate is high, and the TMB estimation will be unreliable. So the TMB estimation has to be done with the false positive rate well controlled. So there are many factors that can influence TMB calculation. For example, if the tumor cell content is low, that means the tumor cells in your sample, the higher sequencing coverage is required, which may also lead to higher false positive rate. Fresh frozen samples are usually preferred, but in many applications, only FFPE samples could be supplied. In such cases, the artifacts and the damage introduced by the FFPE processing need to be taken into consideration. We're actually working on another manuscript looking at the FFPE damages. So as I mentioned, TMB is typically estimated by the WES or WGS in the research study, while the clinical testing prefer the panel sequencing. And how the two estimations are aligned, in another word, how the TMB estimate with the, for example, WES could be translated to the TMB estimated with the panel sequencing need to be addressed. Of course, the wet lab procedures and the downstream analysis may also impact the variant calling and thus affect the TMB calculation. The statistical perspective, all these factors will increase or decrease the sensitivity and the false positive rate, and so the TMB. In this study, we have already learned that the elevated false positive rate outside of the CTR regions will have a strong effect on the accuracy and will have a strong effect to the TMB estimation as well. So this leads to a reasonable requirement that the TMB estimation should be confined to the CTR portion of each panel. The reproducibility is something always in our mind, and for the TMB estimation, we also eager to see how reproducible the TMB estimation could be. So with our data, we have a sufficient number of variants in the known positive set So we computed the average technical run-to-run variance of the panel-focused TMB estimation at the different TMB levels between 5 and 50 per million base through a downsampling approach. 6 panels with targeted region in the CTR greater than 250 kilobases were included in this analysis. The results show that the technical CV decrease at the higher panel TMB values. And all 6 panels achieved a CV lower than the 25% when the panel TMB was 25 per million base or higher. We did this analysis on both the VF cutoff 2.5% and 5%. To assess the magnitude of additional variants introduced by the genomic coverage of oncopanels, we utilized the TCGA WES mutation data to compute the TMB estimation using each panel's CTR portion. We then use the mean squared deviation to measure the TMB estimation difference between the whole exon and the panel-specific CTR portion. We characterize an intrinsic CV measure specific to each panel related to the variance component of the mean squared deviation. So this is our estimation between the TCGA WES and our panel focused region. So a 1 million base pseudo-oncopanel is estimated. And this one have an intrinsic CV of 25% at a TMB value of 21. So using this one as example, because the 1 million base panel is somehow the lowest one reported in some of the studies to be used as a TMB estimation. So we take that one as a reference. And then we put the technical and the intrinsic CV together, and we found that in all the 8 participating panels, adjuvant panel, and combined with a VF cutoff at 5%, would be the only panel for the TMB estimation. And of course, adjuvant panel is also the biggest one among the 8 panels. So in summary, what we have learned in this study, a reference material is very important. Our reference material have a high density of known variants, known positives and known negatives, and in a large range of the allele frequencies. So this kind of the reference materials is very helpful for the assessment of the performance of OncoPanel. So for the sensitivity, all the panels have a good sensitivity, and the spike-in approach is a good approach, but need to be in good use. So we only mainly focus on the SNVs, but for the further study, if we can have more numbers of the small indels and the MNVs, and we can do better estimation on other type of the variants. So to balance the sensitivity and the false positive rate, we think a 22.5% of VF cutoff is the balanced one, and both can have a sensitivity, high sensitivity, and well-controlled the false positive. So to control the false positive rate and focus the analysis on the certain genomic regions, such as the CTR regions we proposed, and the false positive rate will be low, and also you can get good the TMB estimation on those regions. So I would like, this study has taken many years, 4 to 5 years now, and consumed a lot of tangible and intangible resources. I would like to take this opportunity to thank the great SEQC2 OncoPanel sequencing working group and to the leaders, Dr. Joshua Xu from the National Center for Toxicological Research, FDA, Dr. Wendell Jones from the Q-Square Solution EA Genomics and the president of MAQC Society, and also Dr. Leming Shi from the Fudan University, China, who is a long-term leader of the MAQC and SEQC projects. And also, I would greatly thank to all the 151 researchers from 100 institutes, government agencies, and companies in 14 countries who donated their time, reagents, and the computing resources for the completion and analysis of this project. Especially, I want to thank 8 participating panels, companies, for the donation of the samples, reagents, sequencing resources, labors, times, and their thoughts and the continuous support. Thank you all for your attention and the time.
Great, thank you, Binshen, for the wonderful presentation. And we'll now open the floor for questions. So the first question that so please, if you have questions, and please type into the chat box. I have the first question from Charles Wang. What is the overlapping of SNVs among 8 panels? They are from different vendors. And then the second question, were the samples A, B, C derived from cell cultures or prepared using genomic DNA? If cell cultures involved, what is the likely events or effect of genome drifting on the true SNVs for the samples?
Thank you for the questions. The panels actually, well, even though they are OncoPanels, but the overlapping among the 8 OncoPanels actually is small. Of course, if I remember correctly, the biggest panel is the Allele panel, and the panel covers half of the size of the union of the 8. So if you put all the 8 panels together, and the Allele panel can cover half of the union. So, but if you take in the overlap, and actually, for some of the panels, they're actually the size is quite small. So if you take the overlap, it's also very small. The sample A is a mixture of 10 cell lines. Of course, the cell lines are cultured, but they after they made a great amount of the cells, and then they mix them together. So what we got, the sample A is extracted the DNA, and then we distribute the DNA to the vendors. So all the panels should get the same DNA. It's not in different batch of the culture. I don't know if I answered correctly. If you have more things, you can.
So I can add a couple sentences, and Charles. So yes, we do have some concern about genome drifting as well. So so when Agilent prepared the sample, they're going to sequence in the individual cell line and monitor them and making sure that any change to the SMV would be noted. Okay, so we have another question from Dr. Jim Wiley. Is panel performance sensitivity, false positive rate, or reproducibility better for hotspot actionable mutation compared to long hotspot, long actionable mutation?
Oh, I think we didn't directly compare these, but however, the sensitivity is extremely high. So I believe the performance between the hotspot actionable mutation and the non-hotspot should be at the same level. For the false positive rate, of course, they are false. They are not true mutations. They were reported by the each panel because they are not a true variant. So we only think that the region and matters, and we didn't talk about the hotspot or not hotspot for the false ones.
Okay, great. One more question. Did you look at the amount of input of each assay or impact of UMIs to reliability of calling low frequency of VAF?
The question, the answer is no. Even though we have the input, the DNA input amount for each panel are different, but well, if you want to compare the input amount, and you should put, for example, different input for the same panel. But for us, and each panel were fixed because the panel, when they designed the panel, they require a certain amount of DNA input. So we didn't compare that. We leave that to the panel provider, and as what they designed, how much input should be is required is in their panel design. So we didn't compare that. The UMIs, and we have 3 panels using that UMI technology, but they are not for the variant calling. They basically only using the UMI for the deduplication or error correction. So I know that some of the pipelines take advantage of the UMI. For example, they cluster the reads with the same UMI and make consensus, but no, in our study, they didn't. The panels in-house or recommended pipeline didn't take advantage of that one. They only use for the deduplication.
Okay, great. Thank you very much, Binshen.